talking about today? Genetics. Genetics, the study of inheritance. Now, we kind of understand now that inheritance, the inheritance of physical traits that make offspring look like their parents, is based on the transmission of DNA from parent to offspring. So this is a good follow-up to our discussion about meiosis. We talked about meiosis in the previous video. And this determines the patterns by which traits, physical traits that we observe, are passed on from parent to offspring. So here we have a diagram of a cell with chromosomes in that cell. Try and remember the terminology. A chromosome is a molecule of DNA wrapped around its respective proteins, which we call histone proteins, correct? So here's a chromosome. They've magnified one chromosome, and what you'll see here as I fast forward through the presentation, one chromosome will have many different genes on them, right? What is a gene? So try not to mix up the words DNA, chromosome, and gene. Well, what is a gene? A gene is a sequence of DNA that codes for a particular protein. And all of the traits in your body ultimately can be traced back to the work of a protein. The way you look is based on a protein. The way you digest certain foods is based on a protein. And every protein is coded for by one gene, by one sequence of DNA. One of many sequences of DNA coded for on a specific chromosome. So here we have a homologous pair of chromosomes. More terminology for you guys to keep track of. A homologous pair of chromosomes means two chromosomes with the same genes on them. Each of your cells, a human, has 23 pairs of chromosomes. So 46 chromosomes, 46 homologous pairs, 23 from your mother and 23 from your father. And these contain all of the genes that make you, you. So if we look here at this diagram of a homologous pair of chromosomes, we can see that they're using letters to represent the different genes that would be present on a chromosome. Now, a typical chromosome would have many more genes than this, but it's just an example. We see here genes B, C, D, E, all the way to K. And you may notice that some of these genes are represented by two different letters. So here we see two capital B's. Here we see an uppercase C, a lowercase c, uppercase D, lowercase d. Okay. These different versions of the gene are called alleles. An allele is an alternative form of a gene, a version of a gene. What does this mean? It means that you, because you've inherited one chromosome from your mom and one chromosome from your dad for each of your 23 pairs, that you may have inherited, if we go with eye color as an example, you may have inherited the blue eye allele from your father and the brown eye allele from your mother. So we would say that if you have two of the same alleles for a gene on a chromosome, that you are a homozygote. You have two identical alleles at a particular locus. You inherited the same version of a gene from your mother as you did from your father. If you have two different alleles for a particular gene, we say you're a heterozygote. The example here could be you inherited a dark hair allele from one parent and a light-haired allele from the other. You're heterozygous. And we represent that using an uppercase E for one allele and a lowercase e for the other allele. You could say that one allele is dominant and one allele is recessive. Throughout this discussion, please don't forget what you learned in meiosis. What are we seeing here? This is a cell, a representation of a cell with two chromosomes. If you look up here, the two chromosomes go through S phase, so they duplicate. Diploid cell, diploid cell. They go through one meiotic division, and this separates the homologous chromosomes. It takes your paternal chromosomes and separates them from your maternal chromosomes randomly for each set of chromosomes. It doesn't mean one cell has only paternal chromosomes and one cell has only maternal ones. They're going to be mixed between the two, but in this example, we're only looking at one chromosome. What happens? Once the second meiotic division is completed, we have 4 different gametes, either sperm or eggs. These could potentially have different genetic codes, therefore different alleles, which could contribute to an organism's appearance, an organism's genetic code, or what we call an organism's phenotype, the physical expression of their DNA. So we understand that the basis of genetics, the basis of this inheritance, is DNA and how DNA is sorted during the production of sperm and eggs, during the production of gametes. Gregor Mendel, the man credited with a lot of what we use as the basis for our understanding of genetics, he did not know about DNA at the time, but he did understand that there was something physical being transmitted from parents to offspring to make them inherit particular traits, right? Whether you're a plant or an animal, human being or otherwise, you always inherit traits from your parents. You always look somewhat like your parent. So he used the pea plant to study this. Some 24,000 plants, and pea plants, of course, are easy to study. They're easy to grow. Pea plants are easy to grow. They're inexpensive. They have a short reproductive cycle. He could control the pollination. In other words, you can take the pollen from one flower and put it into another flower very deliberately. And using that, he could create a model for genetics that turned out to be consistent with how human beings inherit their particular alleles, right? So this is an important point to make, of course. Scientists cannot do horrible breeding experiments with human beings. You need to use a model organism, and this was an appropriate model organism for the experiments that he wanted to carry out. What else was interesting about the pea plant was that he looked at 7 traits, and these traits were very binary in most of these cases. A pea plant could either produce green peas or yellow peas, for example. It could produce wrinkled peas or round peas, purple flower petals or white flower petals. And this made his experiments very easy because, well, if you look around you at any particular time, especially if you live in Montreal, you'll see there's a wide range of traits. It's not kind of binary. Someone is not just tall or short. Someone is not just, they don't just have brown eyes or blue eyes. There's a wide spectrum. But by studying the pea plant, he had the opportunity to study some very discrete traits that were either one or the other. You'll see here in your notes the word phenotype. Again, the physical expression of your genetic code is called the phenotype. So each of these traits is expressed by phenotypes. There are words here that we'll continue to use: pure breeds and hybrids. What does that mean? A pure breeding organism produces identical offspring when self-fertilized. So you can also self-fertilize plants. So if you self-fertilize plants, you will always get the same traits again and again. Why? Because they have the same alleles for any particular gene. A hybrid is a mixture of 2 different pure lines. So the example that we're going to look at here shows what happens when you cross true breeding purple flowers with true breeding white flowers. And the results of that experiment were as follows. So Gregor Mendel called the parental generation, generation P, and again these were true breeding parents. In other words, if you take these purple flowers and self-pollinate them, they'll always produce purple flowers. If you take these white flowers and self-pollinate them, they'll always produce white flowers. But he crossed them with each other. And he found that in the first generation, all of the plants produced purple flowers. He called this first generation F1, the first filial generation. And he made a very interesting observation. It might be interesting to you. See, there's no kind of in-between trait with flower petal colors in the pea plant. They all became purple, which implied, of course, that one allele was dominant over the other. Self-pollinating the same plants, again from this F1 generation, gave an interesting result as well. It showed that you got mostly purple plants again, but you also got about a quarter of these plants that were white. So we saw the reappearance of what appeared to be a recessive trait. This white flower petal color came back. And you should pay a close attention as well to the numbers here. The numbers are approximately 3 to 1. So, a little review. Homozygote, you have two identical alleles for a particular trait. It could be true breeding purple, which means you're homozygous dominant. We could be true breeding white, in our example, which means you are homozygous recessive for a particular allele. And when you cross the two, you get hybrids, or you get heterozygous organisms that were purple as well. Why? Because the dominant purple allele hid the effect of the recessive white allele. So again, be confident with your terminology. What's a phenotype? A phenotype is what you see. What's a genotype? It's your genetic makeup, and in the context of what we'll be discussing, it's the combination of alleles that we will see for a particular gene. Some dominant traits in human beings include rolling your tongue. We did this in class. It amuses me to watch a whole class of people do this. It especially amuses me to watch people who can't do it and watch the tongue rollers condescend the non-tongue rollers who have two recessive alleles for the tongue rolling gene. It's genetic. You can either do it or you can't. It's not a matter of how much effort you put in. Just like ears, see, I have attached earlobes like Charles Barkley, and which is why we have Barack Obama here with unattached earlobes. He looks pretty happy about it. I wish I had unattached earlobes. Or do I? I don't know. It's okay. This caused Gregor Mendel to come up with two laws. His first law was the law of segregation. And the law of segregation says that two copies of a gene separate when they make individual gametes. In other words, the gametes, as we know from our study about meiosis, gametes are haploid. So if you as an individual are heterozygous for a particular gene, you can only produce gametes that have one of those two alleles. So your gametes, in this case, we have a heterozygous parent who's big R, small r. Here we're saying this is a gene for seed shape. So that's not you. You don't produce seeds. This is R, big R, little r. Your gametes will be one or the other. They can't be both. They cannot be both. They can only be one or the other. Now let's look and see this table here, which we call a Punnett square. And this is going to be a very useful tool for you in solving these genetic problems. So we're looking at a homozygous plant. This is the female plant, and this is the male plant. They're both heterozygous. And because of the law of segregation, we know they can produce gametes that are either big R, little r, big R, little r. When you make a table like this, what you're doing is you're representing all the combinations that can result from two heterozygous parents producing gametes. So you can get an individual's big R, you can get an individual's big R, little r, or you can get an individual's homozygous recessive for the R trait. Punnett squares are very interesting because they also represent a probability. In other words, when you look at this Punnett square, well, you have a 1/4 chance of getting an individual's homozygous dominant and a 1/4 chance of getting an individual's homozygous recessive. Here we have 2 1/4s for the likelihood of producing a heterozygous individual. So 1/2 of all of the offspring of these plants will be heterozygous and have round seeds. And of course, we also see here our 3-to-1 ratio. 3/4 of our offspring are going to show the dominant phenotype. Round seeds are dominant over recessive seeds. And as I said, this relates to what we learned in meiosis, and it's the basis for Mendel's principle of segregation. A human example would be this particular family, Brazilian. They're dark-skinned Afro-Brazilians, and in this family there are albinos. There's an allele for albinism. So this woman had 5 children. Her husband is also dark-skinned, and we see here that some of their children are they're exhibiting this trait of albinism. So you can calculate the probability of this happening. Maybe some of you guys will be genetic counselors in the future. And for a trait that is inherited as simply as albinism, albinism is an example of complete dominance. By that I mean albinism is a recessive allele, so you need 2 copies of the recessive allele to be albino. And you can calculate the probability that 2 individuals might have a child with albinism. So the first question you want to ask yourself, well, if the two parents are dark-skinned, in other words, they don't exhibit albinism, and they have children who exhibit the recessive allele, who do have albinism, can you deduce the genotype of the parents? What combination of alleles do they have? And you would have to conclude that both parents are heterozygous for the gene for albinism. Why? Because each one of them has to give a recessive allele to one of their children. So let's look at that. Let's unpack this problem a little bit. Big A and big A little a, so you can be homozygous dominant or heterozygous. This would give you normal skin pigmentation. You need 2 copies of the recessive allele to show albinism. So, as I said, if 2 individuals are going to give a child with a recessive phenotype, you know that the parents of these children must have 1 recessive allele each. So you would cross those 2 parents. And we'll use a Punnett square to do that. Okay, so what is the probability that these 2 parents will have albino children? We add the genotypes of the parents in this part of the Punnett square here, big A, little a, big A, little a. And this represents gametes that can be produced by, let's say, the mother here. These are the mother's possible gametes. These are the father's possible gametes. And they can come together in this combination. So what do we see here? The probability of a first child getting albinism, the probability of any of their offspring getting albinism, is 1 out of 4. 1 out of 4. Okay, the probability of a child, of one of their children not having albinism is 3 out of 4. 3 out of 4. Right? Mendel's second law of inheritance, and we call that the law of independent assortment. So if you're talking about 2 genes, and a lot of the problems that we're going to be doing in class are going to involve 2 or maybe more genes, and these genes assort independently. There's a condition to this. They assort independently if they are on different chromosomes. So here we have 2 genes, R and Y, okay? And R and Y are on different chromosomes. So when you produce gametes of R and Y, you can get every combination of alleles of the 2 different genes in each of your gametes. So you have 8 possible gametes that you could produce. Well, I should say 4 possible gametes that you could produce, depending on where you're getting your particular chromosome from. And you could do a really large Punnett square to kind of represent this. All right, so if you look here at this large Punnett square, we're looking at the gametes that can be produced by each parent and how they can come together to form different types of offspring. I wouldn't recommend doing these large Punnett squares. I would do small Punnett squares, 1 for each gene, and that's exactly what I'm going to show you how to do next lecture.